Welcome to the AI Research Roundup. I'm Alex. Today we're looking at a paper trending on X, published on December 30th, 2025, about 5 days ago. It introduces the Science Context Protocol, an open-source standard that unifies over 1,600 scientific tools into a single collaborative ecosystem for autonomous agents. The paper is titled SCP, Accelerating Discovery with a Global Web of Autonomous Scientific Agents. And as we'll see later, the platform enables AI agents to autonomously design and execute complex experiments, bridging the gap between computational models and physical laboratories. Alright, Figure 1 illustrates the high-level architecture of the Science Context Protocol. At the very center sits the SCP hub, which serves as the intelligent brain that coordinates the entire system. On the left, you have the various initiators, such as human researchers and AI scientist agents, who submit experimental goals. The hub then connects these requests to the resources shown on the right, which include everything from physical wet lab instruments to digital databases and specialized models. Now, Figure 2 details the specific architectural layers that make this coordination possible. At the very top, we have the SCP clients, which provide the interface for researchers to design experiments and submit requests. Below that sits the SCP hub, the central engine that interprets these requests using modules for advanced planning, authorization, and task assignment. Finally, the hub directs the SCP servers at the bottom, which manage the actual execution resources, ranging from databases and software tools to autonomous agents and physical lab devices. So, having established the architecture, Figure 3 analyzes the actual tools integrated into the intern discovery platform. On the left, the disciplinary breakdown shows a clear focus on the life sciences, with biology-related tools making up nearly 46% of the ecosystem, followed by physics at roughly 21%. Then, looking at the functional distribution on the right, we see that computational tools and databases form the backbone of the system, together accounting for almost 3/4 of all resources. Well, to see these tools in action, Figure 4 visualizes Case Study 1, an automated polymerase chain reaction protocol. The process starts in the top row, where a researcher submits a natural language request, which the Thoth AI agent translates into a structured plan. Then, looking at the bottom row, that digital protocol is converted into atomic commands. These pass through the secure interface to robotic instruments, like automated pipettes, which physically run the experiment and generate the final results. So, while the previous example started with a fresh request, Figure 5 illustrates Case Study 2, automatically reproducing an experiment from a PDF file. The workflow starts in the top row, where the system ingests a document, extracts key parameters like reagent volumes, and reorganizes them into a precise JSON format. Then, the middle row shows how this code converts into an atomic plan, passes safety validation, and triggers the physical lab instruments. Finally, looking at the bottom, a monitoring dashboard tracks the entire process, providing feedback loops for safety and optimization. Now, Figure 7 presents Case Study 4, which takes integration a step further with AI-assisted fluorescent protein engineering. The top row displays the initial computational phase, where agents handle protein design, sequence analysis, and 3D structure modeling. Moving to the bottom row, the pipeline bridges the gap to physical reality. It starts with molecular dynamics simulations on the left, then triggers automated wet lab equipment for expression and purification, and finally delivers the fluorescence characterization results shown on the far right. Alright, expanding on the infrastructure behind those case studies, Table 1 lists some of the key servers currently active in the ecosystem. At the top, the Venus Factory server focuses on biology. It unifies 12 tools for AI-centric protein engineering, covering workflows like mutation prediction and model training. Below that, the Drug SDA tool server offers a broader integrated toolkit. It bundles 33 functions, including OpenBabel and RDKit, to handle tasks ranging from ligand normalization to binding pocket analysis. Finally, the Origin Chembl server at the bottom connects biology and chemistry, utilizing 58 tools to query small molecule bioactivity data. Wrapping up, 2 takeaways stand out. First, the Science Context Protocol unifies over 1,600 fragmented tools under a single open-source standard. Second, by connecting digital planning directly to physical execution, it allows agents to autonomously drive complex experiments in the real world. This framework paves the way for a truly global web of automated discovery. That's it for this episode of the AI Research Roundup. I'm Alex. Thanks for listening.